Dear learners, last module you learned about introduction, sources and the effect of processing. In this module, you shall study the beneficial effects of phytates. Phytic acid is found in cereals, legumes, nuts and oilseed, constitutes up to 1 to 5% of the weight of cereals or oilseeds and serves as the chief storage form of phosphorus. Phytic acid has long been considered an anti-nutrient, mainly due to its ability to bind with many divalent cations, proteins and starch, and to consequently reduce their bioavailability. This binding ability is attributed to phytic acid's highly negatively charged phosphorylated structure. To combat phytic acid's anti-nutrient effects, particularly in nutritionally compromised populations, many ways of removing it from foods have been suggested. However, it has also been suggested by many studies that consumption of phytic acid may convey some beneficial health effects. The possible beneficial effects of food phytates include lowering of serum cholesterol and triglycerides and protection against certain diseases such as cardiovascular disease, renal stone formation and certain types of cancer. This module will acquaint learners with mechanism of phytate hydrolysis in the gut and absorption and physiological effects of phytates. Mechanism of phytate hydrolysis in the gut. Studies in humans showed that 37 to 66% of dietary phytate is regarded during digestion in the stomach and small intestine, when the diet is rich in plant food phytases. In humans, the main phytate hydrolysis occur in the large intestine by means of microbial phytases. Due to the stepwise degradation of dietary phytate in the gut, large number of inositol phosphates are formed. Among the inositol phosphates typically formed by 3 and 6 phytases. Other inositol phosphates such as DL-inositol P4 and DL-inositol P3 are formed. The higher phosphorylated inositol phosphates are the lower the solubility of their metal complexes in the gastrointestinal chyme. Consequently, high phosphorylated inositol phosphates and especially phytic acid bind strongly to minerals and trace elements under the acidic conditions of gastric chyme and form soluble complexes. During the passage from the stomach to the small intestine and the increasing pH, they precipitate. Thus, the availability of bound trace elements and minerals is reduced. The intestinal absorption of these elements affected under certain dietary conditions and imbalanced nutrition. This may lead to serious deficiencies of these elements in humans and animals. Now, let us see the absorption. The phytate absorption in the gut is very low and does not exceed a few percent. Whether phytate or other inositol phosphates might be absorbed via active transport, phagocytosis or other ways of absorption, or by combination of these also remain to be clarified. Next, we will go through the physiological effects. Research has traditionally focused on its structure that gives its ability to bind minerals, proteins and starch and thus lowers absorption of these elements. The first physiological effect is phytate and its mineral interaction. Phytate is a strong chelator of divalent minerals such as copper, calcium, magnesium, zinc and iron. Several in vitro and in vivo studies in animals and humans clearly indicate that phytates decreases mineral bioavailability by forming complexes with these minerals. Many of phytate mineral complexes become unavailable for absorption under normal physiological conditions. It contributes mineral deficiencies in people whose diets rely on these foods for their mineral intake. such as those in developing countries. The effect of phytic acid on mineral availability depends on numerous factors. They are amount of phytic acid present in it, concentration of minerals, molar ratio phytic acid to minerals, size and valency, presence of other metal ions, presence of other substances which can bind minerals, example fiber, tannins, oxalic acid, and even processing methods used. At the pH values between 5 and 7, which are similar to those in the duodenum, phytic acid bind and forms insoluble chelates with divalent cations. At pH 7.4, the order complexion is decreasing order, which is copper, zinc, cobalt, manganese, iron, and the last calcium. Next, interaction with niacin. It also acts as an acid chelating the vitamin niacin, which is basic, causing a condition known as pellagra. This was observed people with a particularly low intake of essential minerals, especially young children and those in developing countries. Next, phytates and protein interaction. Phytates are ionic in nature. They also react directly with charged group of proteins or indirectly with the negatively charged group of proteins, mediated by a positively charged mineral ion such as calcium. The resultant phytate protein and phytate mineral protein complexes may also adversely influence protein digestion and bioavailability. And the next one is phytate and starch interaction. Phytate can also bind with starch either directly by hydrogen bonding with a phosphate group or indirectly through the proteins, which may result in a decrease of starch solubility and digestibility. Now, let us conclude this topic. In this module, we have learned the hydrolysis of phytate, which occurs in the small and large intestine by means of plant or microbial phytases. During the stepwise degradation of dietary phytates in the gut, large number of inositol phosphates are formed. We have also studied the physiological effects of phytates. Several factors are present which govern the inhibitory effect of phytate on mineral bioavailability, pH content of minerals and phytate solubility of phytates, and moreover great variation exists for the affinity of phytate complex with different valent cations, showing increasing binding strength from mono to multivalent cations. Furthermore, different phytate mineral ratios in foods and diets seem to be also important for phytate inhibition of the intestinal absorption of minerals and trace elements. The phytate concentration in the diet has to exceed a certain level to have a substantial effect on the bioavailability of minerals and trace elements.